part two of my overview and review of Ori Hofmekler's Warrior Diet. Before I start, I want to make sure people understand I'm not any kind of doctor. I'm not an MD. I'm not a DO. I'm not an ND. I'm not a chiropractor. I'm not a nutritionist. I'm not a dietitian. I'm not a chiropractor. I'm not a nurse, not an LPN, not an RN, nothing like that. So, in other words, if you have a medical question or concern, find somebody that's licensed to practice medicine. I'm not, okay? So don't throw your medical questions towards me. I'm just a guy on YouTube. So, human implications of caloric restrictions, effects on aging in laboratory animals. Okay, an overview of opportunities for research. So, for more than 60 years, it has been known that restricting caloric intake below ad libitum levels in several species of experimental animals extends lifespan and slows numerous aging changes. Descriptive research on these effects continues to expand and is being extended to non-human primates. However, there has been little research to explore the implications of these findings for the development of interventions to affect human age-related changes and diseases. In 1999, the National Institute on Aging, in collaboration with the National Institute of Diabetes, Digestive, and Kidney Diseases, convened the Caloric Restriction Clinical Implications Advisory Group to consider opportunities for such research. The group included expertise in gerontology, epidemiology, clinical trials, nutrition, metabolism, endocrinology, neuroendocrinology, genetics, pharmacology, and behavioral medicine. It was composed of 6 panels whose reports follow this overview in brief. Okay, so current epidemiological and intervention data, metabolic interventions, anorexiants and neuroendocrine manipulations, physical activity and body composition changes, genetic variability in responses to caloric restriction, effects of lowering caloric intake in non-obese persons. These panel reports were developed before the Calorie Restriction Clinical Intervention Advisory Group's March 1999 meeting, discussed in its plenary session, and modified thereafter. 3 members of the group, and the reports follow the standard format, including background information and recommendations for studies in non-primates, studies in non-human primates, studies in humans, recommendations on methodologic approaches, and recommendations regarding resources and infrastructure. The following discussion of some of the main themes running through the advisory group's considerations and recommendations emphasizes themes that cut across contributions of several panels. Numerous equally important ideas appear in the individual panel reports. In regard to identifying interventions with potential human application, Panel 2 and 3 focused on the approach that complements the increasing interest in learning the mechanisms responsible for the effects of caloric restriction on aging in experimental animals. One way to probe these mechanisms in experimental animals is to examine how interventions that produce one or more of caloric restrictions, physiological or biochemical effects, lowering circulating glucose levels, for example, affect lifespan or age-related changes. Such interventions are referred to in the following reports as calorie restriction mimetic interventions. If caloric restriction mimetic with a known mechanism of action produces effects on lifespan and aging similar to those of caloric restriction, and I'm going to get into the mimetics in a second. This would implicate this mechanism as a likely mediator of calorie restriction's effects. Testing effects of caloric restriction mimetic interventions in animals also provides an experimental therapeutic strategy to explore potential human interventions, examining how they affect lifespan and aging in experimental animals can indicate potential endocrine or pharmacological agents that might be used in humans to produce some or all of calorie restriction's benefits in delaying or preventing age-related diseases. Some caloric restriction mimetics, such as oral hypoglycemic agents, meaning things that lower the blood sugar, have been studied extensively in humans as agents to treat or prevent specific diseases. In such cases, there may be opportunities for human studies to examine other effects on age-related changes. The amount of information derivable from studies on caloric restriction mimetic interventions has increased greatly with the development of an array of technologies to determine effects on expression of a large number of genes. Using such techniques, effects calorie restriction mimetics could be compared with effects of calorie restriction itself, which have recently been studied with such methods as evidenced by the opportunities discussed in panel 5. A second general, okay, so I'm going to get into caloric restriction mimetics. So this is a study that shows the effect of nutrition. I jumped the gun a little bit there into calorie restriction mimetics. So I'm going to be doing that after this. This has to do with diet quality and age-related biomarkers. Keep in mind that caloric restriction with optimal nutrition, the restriction part is only part of the equation. There's also the optimal nutrition part. So what we need to understand is what role does the quality of the diet play? So I wanted to skip the structure of the research because you can read this on your own. So it's well-structured research, but the ketogenic, this is just going over the different types of diets and how they affect the body. The ketogenic diet reduced cardiovascular disease risk by improving cardiovascular disease biomarkers in type 2 diabetes patients and decreasing inflammation. It also prevented brain aging by avoiding the destabilization of brain network and network switching. Network stability, it is represented by the ability of the brain to maintain functional communication between its regions, and when this process is affected, network switching occurs from the large-scale reorganization of the network modules. Moreover, the Mediterranean diet regulated the relationship between genetic risk factors and cognition, protecting participants with specific variants of CR1 gene. Okay, The Modified Alternative Healthy Eating Index was used to assess diet quality in an older Chinese population, considering consumption of fruits, vegetables, soy protein, fish, meat, eggs, whole grain, fried foods, and alcohol. In this study, a high diet quality combined with varied vegetable intake was linked to lower risk of cognitive decline. Daily intake of green tea catechins demonstrated beneficial effects on working memory in adults by decreasing the commission errors in the continuous performance test and time response. Okay, the Mediterranean diet demonstrated beneficial effects in several key points. The role of the dietary inflammatory index used this diet in telomere shortening. An anti-inflammatory diet rich in antioxidants could reduce the rate of telomere shortening by improving the telomerase activity in individuals at high risk of cardiovascular disease. In addition, diets rich in fibers, vegetables, and fruits, and poor in meat. So that you have a lot of fiber, vegetables, and fruit, and not a lot of meat or dairy products, and not a lot of polyunsaturated fats, as well as waist circumference reduction, are beneficial in improving basically white blood cell and telomere length. Okay, so in contrast, consumption of ultra-processed foods and processed meat was negatively associated with Mediterranean diet adherence, and it was positively related with a higher risk of having shorter telomeres. So ultra-processed foods shorten your telomeres. Ultra-processed foods was also linked to increased risk of cardiovascular disease, ischemic stroke, ischemic heart disease, cerebral vascular disease, and all-cause mortality. Cardiovascular risk factors were also attenuated in the presence of a dietary approach to stop hypertension diet. So that's the DASH diet. Decreasing the levels of fibrinogen, which is what makes blood clot, insulin, and diastolic blood pressure, in contrast to a Western-like diet, a Western-like high-fat diet, and a Western diet like high-carbohydrate diet. Mediterranean diet meals promote positive effects on glycemic, insulinemic, and lipidemic responses. Okay, due to lower levels of glucose, insulin, triglycerides, and non-esterified fatty acids, the ketogenic Mediterranean diet supplemented with phytoextracts also led to reductions in weight, body mass index, and waist circumference %. Fat mass, total cholesterol, LDL, a reduction in LDL cholesterol, a reduction in triglycerides, and a reduction in blood glucose, and an increase in HDL cholesterol, which is the good cholesterol. A healthy Nordic diet reduced non-high-density lipoprotein cholesterol, non-HDL cholesterol, low-density lipoprotein cholesterol, a ratio of apolipoprotein, having a beneficial effect on low-grade inflammation. The Southern European Atlantic diet, a common diet in the Iberian Peninsula, enabled reduced concentrations of C-reactive protein, triglycerides, and insulin, which could help prevent myocardial infarction. Diets rich in potassium intake attenuated cardiovascular disease risk, since they prevented cardiomyocyte injury through high-sensitivity cardiac troponin and cardiac dysfunction through N-terminal pro-B-type natriuretic peptide. A restricted calorie diet allied to daily consumption of grapeseed extraction improved blood lipid profile and helped ameliorate cardiovascular disease risk factors by reducing LDL cholesterol, total cholesterol, and triglycerides, visceral adiposity, meaning less fat in the middle, and the plasma atherogenic, vascular, or what do you call it, visceral adiposity means fat around the organs. So in older adults, a low-fat diet rich in fibers improved periodontal disease, meaning it helps with your gums. Okay, so in older adults, a low-fat diet rich in fiber helped with periodontal disease markers such as clinical attachment loss, bleeding on probing depth, and gingival crevical fluid, as well as on metabolic profile, body weight, and CRP, and glycated hemoglobin. A modified version of the Mediterranean diet score, Elderly Dietary Index, which evaluates elderly adherence to dietary recommendations, was associated with reduced weight. So if they were actually following this diet, they had reduced weight, reduced BMI, reduced waist circumference, reduced insulin, reduced fibrinogen, reduced diastolic blood pressure, better homeostasis, better insulin resistance, alanine, fruit consumption both in fruit or in fruit juice, reduced several cardiometabolic factors such as BMI, waist circumference, glucose levels, and LDL cholesterol in elders. I do think, however, that there is enough research on fruit juices that you probably should avoid fruit juices, and really you should avoid fruit in general unless it's like berries. Med diet also decreased hepatic steatosis risk based on fatty liver. A low-fat diet and Mediterranean diet decreased weight, BMI, waist circumference, and triglyceride levels in adults with type 1 diabetes and metabolic syndrome. Mediterranean diet also decreased atherothrombosis markers such as platelet activating factor, acetyl hydrolase, HDL bound. Discussion. The systemic review was accomplished. The diet studied in this systemic review presented diverse similarities. Ketogenic diet is a low-carbohydrate diet, whereas the Mediterranean diet is based on low consumption of saturated animal fat and red meat, and a high intake of fruits, vegetables, fish, olive oil, nuts, and vitamins. The DASH diet pattern is very similar to the Mediterranean diet, since it is rich in the same products and poor in saturated fatty acids, added sugars, sodium, and refined carbohydrates. The maintains the use of vegetables and olive oil, just like the Mediterranean diet, but presents elevated ketone bodies in the blood or urine. The the Southern European whatever that is represented by high intakes of fresh fish, red meat, and pork products, legumes and vegetables, vegetable soup, potatoes, dairy products, whole grain bread, and wine. The Baltic Sea diet is described as a variant of the Med diet with healthy Nordic foods. The healthy Nordic diet is based on the consumption of whole grain products, abundant intake of berries, fruits, and vegetables, rapeseed oil, 3 fish meals per week, low-fat dairy products, and a reduction of sugar-sweetened products. Is a very low carbohydrate diet. The MMKD is a very low carbohydrate diet that is modeled on the Mediterranean diet, emphasizing protein sources such as low in saturated fat like fish and lean meats, healthy fats, fruit and vegetable consumption, whole grains, and a glass of wine per day. The adoption of a ketogenic diet reduced cardiovascular risk by improving cardiovascular disease biomarkers and type 2 diabetic patients, and decreased inflammation, since inflammation has a role in every facet of cardiovascular pathogenesis. And the Mediterranean diet modulated the impact of genetic risk factors on cognition. The variant study the Mediterranean diet used to assess role. Okay. In conclusion, the systemic review demonstrated the necessity for individuals to improve their diets to reduce the emergence and development of several comorbidities and promote healthy aging diets rich in vegetables, fruits, nuts, cereals, fibers, fish, unsaturated fats containing antioxidants, vitamins, potassium, omega-3s, and reducing red meat and, unpolyunsaturated fatty acid intake could prevent obesity, cardiovascular disease, and inflammation, and promote favorable glycemic, insulinic, and lipidemic responses. Moreover, the Mediterranean diet and ketogenic diet, or a combination of these diets, and increasing consumption of vegetables and green tea catechins could improve one's working memory and decrease destabilization of the brain network and the attention domain, preventing cognitive decline. Finally, the Mediterranean diets supplemented with coenzyme Q10 and extra virgin olive oil, or a low-fat, that would be virgin olive oil, or a low-fat diet also rich in antioxidants could help to decrease the prevalence of atherothrombosis, which means blood clots, hepatic steatosis, diabetes, and telomere attrition, meaning the shortening of telomeres, as well as prevent oxidative and DNA damage. These diets can enhance one's quality of life and increase life expectancy. Moreover, a putative panel of molecular markers would follow the impact of diet nutrition alterations during aging. People have, of course, heard me talk about the zone diet if they followed my videos. So the zone diet, I don't know why they gave it such a low score, but the zone diet has been popular for several decades. It encourages followers to eat a certain amount of protein, carbs, fat at every meal, or carbs and fat at every meal. Weight loss 4.5 out of 5. Overall score is only 3.83. Healthy eating 3.75. That's ridiculously low for when you see what the diet is. Sustainability 3.75. Whole body health 2.25. Nutrition quality 5. Evidence-based 3.75. I think it's a little bit better than that, but bottom line, the zone diet focuses on eating a specific ratio of macronutrients to combat inflammation. Though the eating pattern may be linked to several benefits, the proponents of the diet also make many strong and, quote, unfounded claims around its efficacy. What is the zone diet? The zone diet instructs its followers to stick to eating a specific ratio of 40% carbs, 30% protein, and 30% fat. As part of the diet, carbs should have a low glycemic index, which means they provide a slow release of sugar into the blood and keep you fuller longer. Protein should be lean, and fat should be mostly monounsaturated. The zone diet was developed more than 30 years ago by Dr. Barry Sears, an American biochemist. His best-selling book, The Zone, was published in 1995. Dr. Sears developed this diet after losing family members to early deaths from heart attacks and felt that he was at risk unless he found a way to fight it. Now, why am I bringing this up? The zone diet actually allows you to restrict calories pretty substantially without feeling hungry. Okay? By following the higher protein approach that is promoted by the zone diet, you avoid going into ketosis, which can be annoying because it has side effects like the keto flu. And on top of that, it allows you to control your hunger. And you get the benefits of a calorie-restricted diet. So, proponents of the diet claim that once you reduce inflammation, you will lose fat at the fastest rate possible, slow down aging, reduce your risk of chronic disease, and improve your performance. How do you follow the zone diet? The zone diet has no specific phases, again, no phases, and is designed to be followed for a lifetime. There are two ways to follow the zone diet: the hand-eye method. The hand-eye method is the easiest way to start the zone. As the name suggests, your hand and eye are the only tools you need to get started, although wearing a watch is also recommended to keep an eye on when to eat. In this method, your hand takes on several uses. You use it to determine your portion sizes. Your five fingers remind you to eat 5 times a day and never go without food for 5 hours. Meanwhile, you use your eye to estimate the portions on your plate to design a zone-friendly plate. One third of the plate is lean protein, so 1/3 should be lean protein and 2/3 carbs. 2/3 of your plate should be filled with carbs. Now, what that means on the zone diet is fruits and vegetables. A little fat, add a dash of monounsaturated fat to your plate, such as olive oil, avocado, or almonds. The hand-eye method is designed to be a simple way for the beginner to follow the zone. The zone food block is ridiculous. I'm not even going to cover it. I don't know why he just doesn't go with regular calorie counting like a normal person. What foods can you eat on the zone diet? Protein, of course, you can have lean beef, pork, lamb, veal, and game. Skinless chicken and turkey breast, fish and shellfish, vegetarian protein like tofu and other soy products, egg whites, low-fat cheeses, low-fat milk and yogurt. Fat, you can have avocados, nuts, peanut butter, tahini, oils such as canola oil, sesame oil, peanut oil, and olive oil. Carbs, okay, you can have berries, apples, oranges, plums, and more. Vegetables such as cucumbers, peppers, spinach, tomatoes, mushrooms, yellow squash, chickpeas, and more. Grains such as oatmeal and barley. What can't you eat on the zone? High sugar fruits such as bananas, grapes, raisins, dried fruits, and mangoes. High sugar or starchy vegetables like peas, corn, carrots, and potatoes. Refined and processed carbs, bread, bagels, pasta, noodles, and other white flour. Although Dr. Sears does, and now you can get it really easily, but red lentil pasta is something that you can eat on the zone. Other processed foods including breakfast cereals and muffins. Foods with added sugar, such as candy cakes and cookies, soft drinks, neither sugar sweetened nor that's not true. You can have sugar-free drinks. Coffee and tea, keep these to a minimum since water is the beverage of choice. Okay, this is an example of breakfast. So it'd be 2 eggs scrambled, 3 strips of turkey bacon, 1 ounce of low-fat cheese, 1 apple, 3.5 cups of spinach cooked. Now, what's amazing is what you're seeing here is this is a lot of food and a low amount of calories. When you eat high nutrient foods, a lot of times when you eat high nutrient, high fiber foods, you'll be amazed at how much food there actually is and how little calories there actually are. Okay, lunch. 3 ounces of grilled chicken, 1 hard-boiled egg, up to 2 heads of iceberg lettuce, 1 cup of raw mushrooms, 1 cup raw cucumber sliced, 1 red bell pepper sliced, 2 tablespoons of avocado, 1/2 teaspoon walnuts, 1 teaspoon vinegar dressing. Mid-afternoon snack. You're getting the idea here. This is a good bit of food. 1 hard-boiled egg, 3 almonds, 1/2 apple. Dinner, 6 ounces of salmon, okay, 1 cup of sweet potatoes, up to 1 head of iceberg lettuce, 1/4 cup tomato raw, 1 cup raw cucumber, 2 tablespoons of avocado, 2/3 teaspoon of olive oil. Pre-bedtime snack, cottage cheese, nuts, and fruit. As you can see, this is quite a bit of food, and this allows you to restrict your calories while not feeling hungry. So, calorie restriction mimetics against age-associated disease, targets, mechanisms, and therapeutic potential. So, let's get into this real quick. Calorie restriction consists of chronic, meaning long-term reduction of total caloric intake without malnutrition, together with intermittent fasting, which can be regarded as a particular form of calorie restriction in which episodes of ad libitum feeding are alternated with episodes of up to 0 calorie intake. Calorie restriction is the only known strategy to robustly improve health and lifespan in most, if not all, living organisms. In rhesus monkeys, 2 differently designed studies revealed contrasting results on lifespan but similar health benefits and delayed onset of aging phenotypes. In humans, calorie restriction has been reported to counteract several age-associated alterations in non-obese healthy adults. 24 months of continuous calorie restriction was safe, improved the quality of life, and caused 10 to 13% weight loss, reducing fat mass, which stabilized after 1 year. Fasting insulin levels, body temperature, a possible marker of metabolic rate, resting energy expenditure, oxidative stress, and thyroid axis activity were reduced under caloric restriction. Metabolic adaptation, a long-term effective caloric restriction that reduces the metabolic rate below the expected value, occurs in humans and may support longevity. In healthy humans, calorie restriction also decreases the level of circulating tumor necrosis factor, alpha and cardiometabolic risk factors, triglycerides, cholesterol, and blood pressure. Upon calorie restriction and weight loss, insulin growth factor levels and insulin resistance are reduced in obese patients. However, they are not improved in non-obese humans after 1 year weight loss phase. While calorie restriction inhibits inflammation, its effects on immunity need further clarification, since different levels of calorie restriction may subvert and or modulate immune defenses against bacteria and viral infection. Caloric restriction reverses aging-derived effects by initiating numerous pathways involved in the improvement of health parameters: AMPK, AMP, activated protein kinase, cardiovascular disease, mTOR, mechanistic target of rapamycin, tumor necrosis factor alpha, QOL, quality of life. Okay, well, that's what QOL means. Molecular effects of calorie restriction. Macrophagy is a conserved cellular recycling program that eliminates dysfunctional organelles, proteins, and aggregates from cytoplasm, hence protecting cellular functionality and integrity. According to impaired and dysregulated autophagy, has been linked to advanced age, neurodegeneration, cardiovascular diseases, and cancer. In turn, the activation of autophagy via genetic or pharmacological means extends lifespan and or health span in numerous model organisms, including mice. Protein acetylation is a major regulator of autophagy. Okay, and reverse to Interestingly, autophagy and protein acetylation are subjected to circadian fluctuations. This oscillation is lost with aging and has been proposed as a modulatory target of calorie restriction. The maintenance of rhythmic deacetylation by calorie restriction is hypothetically linked to increased NAD+ levels, coupled by SIRT1 activation and rhythmic changes in the inhibitory acetylation of acetyl-CoA generating acetyl-CoA synthase. A short chain family member, blah blah blah, and aged flies. Okay, now we're down to what I want to be down to, which is calorie restriction mimetics. Despite the uncontestable health-promoting effects of calorie restriction, most individuals are unable to observe calorie restriction, likely explaining some failures in observational clinical studies. Although long-term compliance may be improved by periodic fasting regimes, pharmacological approaches that induce autophagy without the subjective discomfort linked to calorie restriction or periodic fasting are warranted. Indeed, several calorie restriction mimetics improve health parameters in rodents and humans. We previously defined calorie restriction as mimetics as compounds that activate autophagy by promoting the deacetylation of cellular proteins. Okay, so the displayed compounds. Okay, mechanistic target. The definition reflects the fact that protein acetylation usually inhibits autophagy, while protein deacetylation favors autophagy. For instance, starvation is coupled to the inhibition of acetyltransferase due to the depletion of ACCOA, as well as to the activation of deacetylase SIRT1. Interestingly, let's see here. So this is a classification of numerous protective substances that are calorie restriction mimetics. So aspirin, hydroxycitric acid, which can be found in diverse tropical plants including Garcinia cambogia and hibiscus. Resveratrol, which is found in fruits, plants, and the skin of grapes. Spermidine, which is found in wheat germ, soybeans, and nuts. Which is a synthetic one. Acetamox, which is synthetic. Berberine, which is found in Berberis vulgaris and several other plants, roots, and bark. Caffeic acid, which is found in eucalyptus bark. Catechin, which is found in cocoa, tea, and red wine. Curcumin, which is found in Curcuma longa. Epicatechin, which is found in cocoa, tea, and red wine. EGCG, which is found in green tea. And these are the different metabolic pathways that it works on. So epicatechin works on these different ones as well. Metformin, which is a synthetic one. Oh, apparently it is present in French lilac, but it's AMPK, mTOR. Okay, these are the different things. NAD+, various food sources. Nicotinamide, various food sources. Nicotinamide mononucleotide, various food sources. Nicotinamide riboside, various food sources. Another synthetic one. This is from passion fruit seeds, but also quercetin. Black tea, so quercetin works on SIRT1. That's found in black tea, onions, rocket coal, curcuma, and fruits. Rapamycin, that's a bacteria, or it comes from a bacteria, I should say. Others, PK. So resveratrol and other SIRT1 activators, the polyphenolic and phytoalexin that is particularly abundant in the skin of grapes and in red wine. It has been shown to promote longevity across species and improve age-related parameters in mice. However, resveratrol seems to only prolong the lifespan of mice on a high-fat diet, but not on a regular chow. Still, resveratrol exerts a number of protective effects. The type 2 diabetes, an effect that enhances decay. However, contrary findings have been reported recently on its efficacy against metabolic syndrome. Okay, resveratrol targets a number of stress-related cellular components. Spermidine is a polyamine that induces autophagy in different model organisms, and this induction is causal for at least some of the observed beneficial effects. For instance, genetic ablation of autophagy and aborates. Spermidine-mediated lifespan extension in yeast, nematodes, and flies, and attenuates cardioprotective effects. In mice, spermidine inhibits the activity of several acetyltransferases. Spermidine is naturally produced in the body by cellular biosynthesis as well as by the intestinal microbiota. In addition, oral ingestion of spermidine contained in food, like wheat germs, soybeans, and nuts, others results in a good bioavailability. Clinical trials are needed to explore, hydroxycitrate acid, depleting it, let's get down to... So the non-steroidal anti-inflammatory drug acetylsalicylic acid, an anti-inflammatory drug, has been in extensive medical use since 1899. Before that, salicylics from white willow bark were widely used in folk medicine. Salicylic inhibits by competing with So, as continuous intake is associated with lower tumor recurrence and decreased metastases. Polyphenols in general, phenolic compounds, may represent an attractive source of calorie restriction mimetics. Epidemiological studies have linked an elevated intake of polyphenol-rich food, mostly fruits and vegetables, and drinks including coffee, tea, and wine, to reduced incidence of malignant cardiovascular and neurodegenerative diseases. Caffeic acid, gallic acid, AMPK activate, so they activate AMPK, protein deacetylation, extended longevity across species, So keep in mind, this is one of the reasons that vegans may live longer than non-vegans, because of the high phenolic compounds in their diet. A number of polyphenols are known to act as inhibitors associated with autophagy induction and health-promoting effects. These include anacardic acid from the nutshell of the cashew and garcinol. Several flavonoids and multifunctional and highly bioactive phenolitic subclass, which comprises more than 5,000 plant-derived substances, also promote autophagy coupled to protein deacetylation. Now, EGCG is interesting because it's unique in the sense that it only comes from green tea. EGCG bioavailability is rather poor, challenging the utility of its clinical use. Metformin is not important, I don't care. Modulation of glucose metabolism. Blocking cellular energy utilization, specifically glycolysis, has been devised as a calorie restriction mimetic strategy. Glucose deprivation alone is sufficient to induce autophagy via AMPK and mTOR. This is one of the reasons that ketogenic diets are protective against a number of things. Conclusion: ongoing and future clinical trials as well as meta-analyses will ultimately determine the actual beneficial impact of calorie restriction mimetics. What's the science behind calorie restriction mimetics? Researchers are exploring the theory that calorie restriction promotes a healthy lifespan by triggering changes in one or more biological pathways identified as playing a role in healthy aging. Biological pathways are a series of chemical reactions that occurs inside the body's cells, much like a production line in a factory. Calorie restriction mimetics research is a search for potential therapies that target the same pathways affected by calorie restriction with the goal of finding ways to extend human health span, defined as how long people live in good health, key biological pathways, insulin signaling pathway, mammalian target of rapamycin, sirtuin-1 pathway, AMPK pathway. With calorie restriction, activity is reduced along the insulin signaling and mTOR pathways and increased along SIRT1 and AMPK pathways. Researchers are looking for compounds that have similar effects to reduce or increase of these activities. Which compounds are most interesting to researchers? Resveratrol, rapamycin, and metformin. Resveratrol is a naturally occurring compound found in red wine and grapes, dark chocolate, blueberries, raspberries, and nuts. It activates SIRT1 and other sirtuin pathways. Animal studies with resveratrol have demonstrated antioxidant, anti-inflammatory, and anti-cancer effects. However, the evidence for human studies is weak. More research is needed to answer questions about whether certain dosages or formulations might promote healthy aging in humans and which populations might benefit. Rapamycin is a substance produced by bacteria found in soil. This compound reduces signaling along the mTOR pathway. Rapamycin has been shown to extend lifespan in mice and to have other beneficial effects. Metformin is a diabetes drug. Blah, blah, blah. Okay. How will calorie restriction mimetics be used in healthcare? If and when calorie restriction mimetics are proven to extend healthy lifespan, they will probably be most useful for people who are unhealthy and unable to eat properly. For people in good health, sensible habits may be more effective at extending health span than taking a medication. So what about polyphenols as caloric restriction mimetics? Okay. Neurogenesis, the complex process by which stem cells in the hippocampus region of the brain differentiate and multiply into new neurons and other resident brain cells, is known to be affected by many internal and external factors. Neurogenesis plays a critical role in neuroplasticity, brain homeostasis, and maintenance in the central nervous system, and is a decisive factor in maintaining cognitive function and repairing damaged brain cells affected by aging and brain damage. Internal factors such as aging, neuroinflammation, oxidative stress, and traumatic brain injury, as well as lifestyle factors such as a high-fat and high-sugar diet, alcohol, and opioid addiction can adversely affect neurogenesis in adults. On the contrary, it has been shown that many dietary components such as curcumin, resveratrol, blueberry polyphenols, sulforaphane, savionic acid, polyunsaturated fatty acids, PUFAs, and diets enriched with polyphenols and PUFAs, as well as caloric restriction, exercise, and training, induce neurogenesis in adult brains. Although many of the mechanisms by which nutrients and dietary factors influence neurogenesis in adults have not yet been determined, nutritional approaches provide promising perspectives for stimulating neurogenesis. So let's look at this again. Remember, eating a healthy diet does matter. This goes back to the quality of your diet. So, of course, eating bad for you foods has a bad effect. But it has been shown that many dietary components, such as curcumin, resveratrol, so fruits and vegetables, blueberries, polyphenols, sulforaphane, savionic acid, polyunsaturated fatty acids like in canola oil, and diets enriched with polyphenols and polyunsaturated fatty acids like in canola oil and corn oil, as well as calorie restriction, which means eating less, exercise and training, so exercising and training induce neurogenesis in adult brains, although many of the mechanisms by which nutrients and dietary factors influence neurogenesis in adults have not yet been determined. Nutritional approaches provide a promising perspective for stimulating neurogenesis. So keep that in mind. We know it works, they just don't know why it works necessarily. In fact, the neuroprotective capacity of natural polyphenols has been extensively studied and reported. One of the most studied polyphenols in terms of neuroprotection activity is resveratrol. Recent study, Lin and co-workers scrutinized the neuroprotective potential of the resveratrol on the rotenone induced oxidative stress cellular model. The data of the study indicated that the treatment with resveratrol suppressed the formation of reactive oxygen species, apoptosis induction, and at the same time increased the survival rate of the neurons. Resveratrol administration led to an increase in autophagic induction and autophagic flux. The study showed, which means it was getting rid of the garbagey parts of the organelles and things like that. The study showed that the neuroprotective effect of resveratrol was a result of the modulation of mitochondria dynamics and upregulating autophagic flux via the MEK extracellular signal regulated kinase signaling pathway. The effect of resveratrol on animal brain affected by the combination of Alzheimer's disease and diabetes mellitus was reported. The results demonstrated that resveratrol significantly elevated sirtuin-1 expression, suppressed the memory impairment, increased the levels of acetylcholinesterase, interleukin-1b, and interleukin. So there is accumulating evidence of an association between the development of Alzheimer's disease and disrupting autophagy mechanism in the affected brain. So re-inducing proper autophagy may help the brain that is suffering from Alzheimer's. Okay. Apart from resveratrol, other natural polyphenols have been intensely explored for neuroprotective activities the last decades, including curcumin, apigenin, quercetin, and apigallocatechin gallate. However, it must be noted that natural polyphenols undergo significant chemical transformations after oral consumption. Moreover, the bioavailability of dietary polyphenols, but remember from my videos on quercetin, part of those transformations in the gut might have to do with the bioavailability and the use of those polyphenols. The multi-component secretion of a huge variety of proteins, including soluble signaling factors, proteases, insoluble extracellular matrix proteins, and non-protein components, collectively referred to as senescence-associated secretory phenotype, is the key feature of cell senescence induced by replicative exhaustion or stressors. Recent data from several laboratories have revealed that senescent cells accumulated with age resulted in an increase in SASP activity in tissues, which would be, again, that's senescence-associated secretory phenotype. Okay. Polyphenols are known to be able to activate autophagy through various mechanisms, one of which is activation of SIRT1 protein by polyphenols based on the stabilizing SIRT1/peptide interactions in a substrate-specified manner. SIRT1 can trigger autophagy by activating autophagy-related proteins. Okay, mechanism is based on the activation of FOXO1. On the other hand, SIRT1 also plays an important role in the regulation of SASP by epigenetic mechanisms. It was established that SIRT1 suppressed the expression of IL-8. Okay, several studies have found out that autophagy induction by multiple stimuli is independent of activation of the inhibitor. Okay. Regulation of SASP and autophagy by polyphenols. There is growing body of evidence that polyphenols can inhibit SASP in senescent cells. Have demonstrated that resveratrol treatment reduced SASP in senescent human fibroblasts. Another study found that resveratrol decreased senescent-associated secretory phenotype by SIRT1 NF kappa B pathway in gut and of the annual fish northo whatever. This polyphenol is also able to serve as an autophagy modulator in senescent cells. Thus, it was demonstrated that resveratrol reduces hydrogen peroxide-induced aging through the activation of autophagy in human umbilical vein endothelial cells. It has been reported that mitochondrial division can occur in an asymmetric manner. This results in the formation of one functionally complete mitochondria. This mechanism demonstrates, autophagy can be stimulated through different mechanisms and chemical agents. In this case, natural polyphenols such as resveratrol can be successfully employed to induce autophagy and thus to improve mitochondrial function. Another polyphenol, which is apagallocatechin 3-gallate, or EGCG, has been extensively studied in context of possible antioxidant activity and improvement of mitochondrial function. The reports indicate that EGCG is able to enhance mitochondrial fat utilization and reduce adipogenesis, meaning the creation of fat tissue. It was also found out that EGCG activates mitochondrial biogenesis, meaning the creation of more mitochondria. Modulation of autophagy and apoptosis by polyphenols. Autophagy can be triggered by different stimuli, including starvation, chemical and mechanical stresses. In fact, calorie restriction has been considered as the main physiological inducer of autophagy. It was found out that the inhibition of autophagy prevents the effect of anti-aging calorie restriction. So it seems that autophagy may be the mechanism by which calorie restriction works, as I went over earlier. Believed to induce autophagy through AMPK activation in SIRT1, which are conjugated and involved in the so-called positive mutual activation loop. How SIRT1 triggers autophagy is not entirely clear. Autophagy plays an important role in organelle protein homeostasis. This role is especially important in non-proliferating cells because, unlike cells in the mitosis phase, there is no dilution of intercellular debris during division. In addition, the anti-aging effects of cytoprotection are especially important for cells that are not targets of stem cells. Aggregation of intracellular proteins, or extracellular junk, or intracellular junk in that case, are distinctive features of many neurodegenerative diseases called proteopathies, including Alzheimer's, Parkinson's, and Parkinson's. It is known that a number of experimental autophagy inducers, such as rapamycin, rapalogs, valproate, and lithium, can weaken the accumulation of mutated protein and reduce the possibility of cell death. Autophagy can increase the body's viability by inhibiting cell death, reducing the risk of oncogenic transformation, meaning the creation of cancer, hormesis, both in dormant and dividing cells. In addition, autophagy can contribute to an increase in longevity through various mechanisms. In the range of studies, it has been demonstrated that caloric restriction improves longevity and adjourn development of age-related disorders. It was also known that caloric restriction triggers many molecular processes. Utilization of redundant products of cell metabolism. Apart from caloric restriction, autophagy can be induced by mechanical and chemical stress. In this regard, various pharmacological compounds have been proposed and studied. This approach facilitates the control and safety of autophagy induction and minimizes the suffering of patients from the strict diet and fasting. In fact, natural polyphenols demonstrated a low toxicity and absence of severe adverse effects. So keep that in mind. Caloric restriction has side effects. Natural polyphenols demonstrated low toxicity and the absence of severe side effects. Moving on. So calorie restriction mimetics and healthy aging in the Okinawan, for Okinawans. So calorie restriction priority for the U.S. National Institute on Aging. However, little is known regarding health effects in humans. Some calorie restriction-related outcomes, such as cause-specific mortality and lifespan, are not practical for human clinical trials. Therefore, epidemiological data on older Okinawans who experienced a caloric restriction-like diet for close to half their lives are of special interest. The nutritional data supported that mild mild caloric restriction, 10 to 15%. So not ridiculously, aggressive caloric restriction, and a high consumption of foods that may mimic the biological effects of caloric restriction, including sweet potatoes, marine-based carotenoid-rich foods, and turmeric. Phenotypic evidence is consistent with calorie restriction, including short stature, low body weight, lean body mass index, less age-related chronic disease, including less cardiovascular disease, less cancer, less dementia, and longer lifespan. Both caloric restriction and traditional Okinawan functional foods with caloric restriction mimetic properties likely had roles in the extended health span and lifespan of the Okinawans. Nutrition is among the most important. Remember, the quality of your diet. Now, I'm going to continue, after I read this article, I'm going to continue with a third installment of this about the importance of the triage theory of aging and disease, the need for micronutrients and amino acids so that people reach the milestones they're supposed to reach and have healthy aging because of the nature by which nutrients are triaged to keep the body functioning. And I'll do that in the next video. Nutrition is among the most important means of mitigating age-associated chronic diseases. By some estimates, 80% of coronary heart disease and type 2 diabetes and 40% of cancers may be prevented by modifying dietary habits, engaging in regular physical activity, and avoiding tobacco use. Highlighting the importance of nutrition, a recent large collaborative 20-year study found that dietary factors accounted for 26% of the risk for early death, the majority and the most disability-adjusted life years lost in the U.S. The Okinawans are of special interest to this topic since they, by most measures, have the world's longest-lived population, though not anymore. It's been replaced by some other area in Japan because of their adoption of a Western-style diet. So the chronic disease profile of the older Okinawan population is especially impressive, with 80% less chronic disease cardiovascular heart disease mortality, and 80% less cardiovascular heart disease and 40% less cancer mortality than the U.S. population, and mirrors the estimates thought preventable by the World Health Organization and the Center for Disease Control. So remember, these are preventable deaths. 80% less cardiovascular heart disease, 40% less cancer. These are estimates that are mirrored. The estimates of the World Health Organization and the Center for Disease Control are mirrored in these statistics. These are preventable deaths. In the U.S., there is 80% more and 40% more. So 80% more cardiovascular heart disease and 40% more cancer deaths. If people would follow an Okinawan-style diet, and engage in more physical activity, you might see these reduced to the same rates. That's why I encourage people to read the Blue Zones and blah, blah, blah. Some attribute the healthy aging phenomenon in Okinawa principally to nutritional factors, and caloric restriction is thought to be one key factor. Caloric restriction, also known as dietary restriction, is a well-known research intervention for those who investigate the biology of aging in model organisms, and is a priority research focus for the U.S. National Institute on Aging, yet few data exist in humans. The most extensive analysis of caloric restriction phenomenon in Okinawan was based on 5 decades of nutritional, phenotypic, epidemiologic, and demographic data. This analysis utilized dietary surveys and population health data from the 1950s to the 1990s to demonstrate that the adult population appeared to be in a relative energy deficit consistent with caloric restriction until the late 1960s, eating approximately only 11% fewer calories, 1,785 kcals per day, than would normally be recommended for the maintenance of body weight. Metabolic adaptations to such conditions included low body weight, shorter height, and low body mass index. Adult BMI was a lean 21 kilogram. Peak body weight was reached in young adulthood and was relatively stable until elderly years. consistent with caloric restriction effect. Prevalence of age-adjusted mortality for particular age-related diseases, especially cardiovascular diseases, cancer, and dementia, is low in the elderly Okinawans compared to other Japanese or Americans, and life expectancy at older ages is long. Life expectancy from age 65 may be the longest in the world at 24.1 years for females and 18.5 years for males. This compares favorably to the 22.5 years and 17.6 years for the same birth cohort in mainland Japan, and 19.3 years and 16.2 years for corresponding U.S. birth cohorts. Despite these data, there is still much controversy about the potential merits of human caloric restriction, blah, blah, blah. This is about the nutrient sensing pathways. We've already gone through this, but the biological basis of the calorie restriction phenomenon is not yet known, but may be linked to nutrient sensing biological pathways which modulate growth and biological systems. Human caloric restriction research has become an area of growing interest in the gerontological research community. The question of whether or not calorie restriction can delay incident age-associated diseases and extend lifespan in humans is a major unanswered question. Studies under the Okinawans have long held that the attention of caloric restriction researchers, since they are perhaps the best human example of naturally calorically restricted population. Okay. Caloric restriction mimetics, and this is what I want to talk about. So these are functional foods. While there are several nutritional factors that are implicated in Okinawan longevity, in addition to caloric restriction, another nutritional factor is receiving increasing attention is the health properties of particular foods from the traditional Okinawan diet. Calorie restriction mimetics, or compounds that provide the physiological benefit of caloric restriction without the need for restriction of calories, are under active investigation. Okay, so let's see here. The traditional Okinawan diet is a rich source for potential calorie restriction mimetics. In the Okinawan language, the term nuchi gushu, a term in common use, literally means food is medicine. Since commonly consumed dietary items, including foods, herbs, and spices, are also used as folk medicines, popular items that play dual roles as foods and traditional medicines are sweet potatoes, pulp, skin, and leaves, bitter melon, turmeric, ginger, mugwort, peppers, and carotenoid-rich marine foods, among others. It is curious that that in Okinawa so many food items might be thought to have medicinal properties, but there may be a scientific basis for this climate. Compounds that have potential caloric restriction mimetic properties, such as carotenoids, flavonoids, and other phytochemicals, are synthesized by plants to help scavenge free radicals formed due to stress from extremes. So it's their hormesis, the plant's hormesis, adaptation to the stress, that creates these chemicals by its reaction to heat, cold, or extreme UV light. Since the sun in Okinawa is particularly strong, many locally grown plants contain high quantities of these phytochemicals. Bacterial and viral pathogens, insects, physical injury, and or other stressors also trigger the production of such compounds. Okay, so typical food items from Okinawa and compared them to typical food items from mainland Japan. Foods from Okinawa have, on average, stronger free radical scavenging properties. Of 138 food items they tested or anti-inflammatory action, many phytochemicals were promising anti-inflammatory agents. Okay, the most common compounds such as polyphenols, flavonoids, terpenoids, strong antioxidant properties. Hormetic properties, so hormetic phytochemicals, meaning phytochemicals that induced hormesis, have received great attention for their potential pro-longevity effects. Some promising functional foods and food compounds from the traditional Okinawan diet are reviewed below. Sweet potato. Okay, the sweet potato has formed the bulk of the Okinawan diet, accounting for well over half of the caloric intake. The sweet potato, we know about the sweet potato. Marine-based carotenoid-rich foods. I'm going to go back over these when I start my next video. More in depth. Turmeric. The clinical implications, should we restrict our calories, blah, blah, blah. So I'm going to go over these. The conclusion, the traditional diet of the Okinawans, who by many measures are the world's longest lived people, provides many clues for research on nutritional aspects of healthy aging and longevity, blah, blah, blah. The Okinawans demonstrate. So I'm going to go over those key foodstuffs in my next video on the quality of the diet. Okay. And this is the second video for Ori Hofmekler and his warrior diet.